Hello everyone, this is Nikhil Malan, and I'll be going over the adaptive immune system. Before we go over the adaptive immune system, I quickly wanted to cover the coagulative system. We all know how we stop bleeding after some time. This is due to the work of the coagulation system. Here, we see a normal blood vessel. Let us analyze what happens when it gets ruptured. Because the blood vessel has been injured, blood can flow out. To stop this from happening, nearby blood vessel cells send out signals known as tissue factor, which is a molecule. These signals are called DAMPs, or damage-associated molecular patterns, which tell all the cells nearby that the blood vessel has been harmed. Platelets and fibrin bunch together to form a clot over the damaged tissue. Platelets are created by other cells in the blood, known as megakaryocytes, and fibrin is always in the blood. The tissue also releases a molecule called bradykinin when injured. Bradykinin first constricts the blood vessel so that the clot can properly form. After some time, bradykinin causes the vessel to dilate so that the immune cells in the blood can flow into the wound and help it heal. In the process, they activate pain receptors in the body and cause pain. Many molecules are involved in protecting our body from pathogens. Most of the proteins are covered in the complement system. However, there are certain molecules that are especially important, that are generally not covered in the complement system because they're not proteins. These are considered advanced innate immune defenses. We'll talk about a few of these. A major part of the innate epithelial protection are defensins. Defensins come in two basic subclasses, alpha defensins and beta defensins. Alpha defensins are secreted by neutrophils and special cells in the small intestine. The small intestine, by the way, has small hair-like organs called villi. The trenches of these villi are called crypts. The cells in the crypt release alpha defensin molecules. Beta defensins are similar to alpha defensins, but secreted in the respiratory tract, urogenital tract, and dermal epithelium. They are made from sections of DNA that are highly mutable, thus making every person's beta defensins slightly different. This ensures a fresh epigenetic profile for the defensin molecule. Furthermore, epigenetics can also cause more or less expression of beta defensins in people, leading to diversity in types and amount of beta defensin. Another important molecule is the alpha-2 macroglobulin. The initial line of defense against pathogens is the complement system, which is a part of our innate immunity. To counter the complement system, which consists primarily of multiple proteins, a pathogen may release proteases that cleave proteins around the pathogen. In response, a molecule known as alpha-2 macroglobulin attracts proteases by displaying a thioester bond, which acts as a trap. Once the protease attaches itself to the macroglobulin, the molecule folds around the protease and signals for macrophages to destroy the macroglobulin and protease. Macrophages primarily function through phagocytosis and are therefore known as phagocytes. Macrophages devour pathogenic material and apoptotic debris to keep the body clean. The macrophages use multiple PRRs, or pathogen recognition receptors, to identify pathogens that need to be phagocytosed. This method of phagocytosis is also known as receptor-mediated endocytosis, since receptors facilitate the phagocytosis of pathogens into the macrophage. One function of macrophages is to phagocytose opsonized pathogens that have already been taken over by the complement system. For the IC3b ligand, the receptor found on the macrophage is the C3R or the C4R receptor. These receptors recognize IC3b and lipopolysaccharides. Pathogenic membranes, or bacterial membranes, often have negatively charged molecules, such as lipids, phosphates, sulfates, and nucleic acids. These are recognized by the scavenger receptor. Mannose, which is a type of sugar, and glucan, which is a type of fiber, are also found in pathogenic membranes, and are recognized by the mannose receptor and the glucan receptor, respectively. One of the most important PRRs is the TLR, or toll-like receptor. TLRs have a conical shape at one end of the receptor, known as the pathogen recognition domain. This is the part of the receptor that actually recognizes pathogenic molecules. The other part of the receptor is known as the signal transduction domain, and is responsible for telling the macrophages that a pathogen has been detected. TLRs result in the upregulation of multiple cytokines, including IL-6, TNF-alpha, IL-1 beta, CXCL8, and IL-12. IL-6 induces hepatocytic protein production, which is a fancy way of saying that the liver cells start producing more proteins. In the context of the immune system, the liver is responsible for producing complement proteins. Thus, IL-6 causes the complement system to gain impetus once pathogens have been detected, allowing for a stronger innate immune response. TNF-alpha stands for tumor necrosis factor alpha. This is an important cytokine because it causes vasodilation, which is an important part of the inflammatory response. Vasodilation allows for immune cells to enter the inflamed tissue. However, excessive production of TNF-alpha can also cause systemic shock because it can cause excessive vasodilation around the whole body, not just in the localized part of the body. IL-1 beta activates lymphocytes and gets them to proliferate into the tissue, and also enhances IL-6 production, which in turn creates more complement protein. CXCL8 is a chemoattractant that communicates with neutrophils and other phagocytes to help them enter the inflamed tissue from blood vessels. IL-12 activates natural killer cells if the infection is caused by a virus and requires the killing of infected cells. When inflammation occurs and the complement system is activated, we need neutrophils to flood the wound and kill the pathogen before it spreads. Anaphylatoxins, released from the inflammation, make their way to blood vessels and trigger the synthesis of selectin and integrin. These proteins are then expressed on the endometrium of the blood vessel. The endometrium is the inside of the blood vessel. The selectin works to slow down the neutrophils in the bloodstream. SLEX receptors on the neutrophils interact with the selectins and make the neutrophil slow down near the site of the inflammation. Eventually, the LFA1 receptor on the neutrophil interacts with an integrin molecule, completely stopping the neutrophil. CXCL8 that originates from the inflamed site signals the neutrophil to perform diapedesis. Diapedesis is the process by which neutrophils retrofit themselves to squeeze through the blood vessels and enter the inflamed tissue. When neutrophils do that, they actually break down their cytoskeleton to become thinner, and using proteases break through the fibers under the blood vessel and get into the tissue. Inside many phagocytes, an important part of killing phagocytosed pathogens is using strong chemicals to lyse the bacteria, so that they don't pose any further threats to the organism. In this case, neutrophils engulf pathogens using receptor-mediated endocytosis, which is basically a fancy way of identifying and eating pathogens. This forms a membrane around the pathogen, called a phagolysosome. Eventually, azurophilic granules, which are membranes filled with digestive proteins and defensins, merge with the phagolysosome and completely decimate the pathogen, thus protecting the body. For most of the time, we've been talking about bacterial pathogens. However, viruses can be just as lethal. Our body has multiple systems to cope with viruses as well. However, interferon is one of the most essential. As you may have noticed in past slides, most bacterial pathogens are phagocytosed by phagocytes before they can do much corporal harm. However, this makes the elimination of viruses a rather daunting task for the immune system. Whilst viruses can exist out of the cell, part of their life cycle is intracellular. To reproduce, viruses take over a host cell and use its transcription machinery to create more copies of the virus. Thus, phagocytes cannot actually target viruses efficiently. Instead, interferon acts as the method for virus elimination. When a virus initially enters a host cell, IRF3 is activated. IRF3 stands for interferon regulatory factor 3 and is responsible for activating the transcription of interferon beta. Interferon beta is a paracrine molecule and affects the very cell it originated from. The IFN beta receptor, upon detecting interferon beta, activates IRF7, which stands for interferon regulatory factor 7. IRF7 activates the transcription of interferon alpha, which is autocrine. Interferon alpha and interferon beta both work to recruit natural killer cells. To understand how interferon alpha and interferon beta recruit natural killer cells, it is important to understand how natural killer cells and macrophages interact. When a macrophage detects pathogenic material in the body, it releases interleukin-12 and tumor necrosis factor alpha. These two cytokines activate and recruit NK cells. NK cells then release interferon gamma, which in turn activates macrophages. Thus, whilst a pathogen exists in the body, macrophages and NK cells continually enhance each other. When an infected cell sends out interferon alpha and interferon beta signals, the natural killer cell is snapped out of the enhancement loop with macrophages and actively begins killing infected cells. At this time, I would like to discuss a huge part of the immune system, which is the B cell. Typically, a B cell begins as a committed B cell progenitor and ends up as a mature plasma cell. A committed B cell progenitor is basically a cell that has decided it is going to become a B cell. A mature plasma cell, on the other hand, is activated when the receptor on the B cell matches a pathogen that is currently in the body. The B cell begins producing antibodies that can attach to the pathogen and help T cells. Antibodies are proteins that attach themselves to certain specific types of molecules, called antigens. Specifically, antibodies attach to a region of the antigen called the epitope. These antibodies come in 5 distinct flavors: IgE, IgD, IgA, IgG, and IgM. IgE is responsible for activating mast cells, which in turn release histamine. Histamine is responsible for vasodilation and the enhancement of the inflammatory response. IgA is useful in neutralizing the parts of a virus that can cause damage. As a dimer, IgA is able to cross mucosal surfaces and is the only antibody found outside of these surfaces. The IgA monomer can also pass into the bloodstream. IgG is the most persistent antibody and stays after an infection the longest. It can also neutralize viruses. IgG is a great opsin and allows for C1 proteins to attach and create C3b. Please review the complement video for a refresher of how C1 helps remove pathogens. IgG can also move across the placenta during pregnancy and is also capable of moving into the bloodstream. IgM creates a pentamer that can pull pathogens out of the bloodstream so as to stop the spread of infection. Now, let us also look at how antibodies are labeled. The 2 prongs of the antibody that actually attach to the epitope are called the variable region, because these are different for all antibodies. However, the rest of the antibody is called the constant region, because it is the same between antibodies of the same type. The 2 chains that go through the middle of the antibody are called the heavy chains. The 2 small side chains attached to the heavy chains are called light chains. Here is the IgA dimer that can pass through the mucosal surfaces. It can be found in the respiratory, digestive, and urogenital tracts. It is an important defensive antibody covering the outside of our epithelial surfaces. The IgM pentamer is also displayed here. Notice how massive it is. When the IgM pentamer attaches to the pathogen, it pulls it out of the bloodstream simply by the increased mass. The pathogen can then only wait as the immune system acts upon it. Our body has a large B cell population. Every B cell has a receptor-antibody pair that is unique to a given epitope. How does our body guarantee so much variation in the receptors and antibodies? This is a question answered by genetics. Let us see how antibodies are formed for a new B cell. This is a visual of our germline DNA and consists of multiple sections. These sections all code for the heavy chain. The first is the variable section with about 40 choices. One of those will be chosen for the new receptor. One of the 23 diversity regions will be chosen, and one of the 6 joining regions will also be chosen. The constant region determines the type of antibody, which is either IgE, IgA, IgD, IgG, or IgM. The first event that occurs is called DJ recombination. This is when a diversity and joining are chosen and joined together. The second event that occurs is called VDJ recombination. This is when a variable region is chosen and attached to the DJ section that was created before. Finally, the introns are spliced out and the mRNA is created. There's a similar process that creates mRNA for the light chain. You can see these displayed here. The mRNA chain on the left is for the heavy chain, and the mRNA chain on the right is for the light chain. This mRNA for the light chain doesn't actually have a diversity region because the light chain doesn't actually have that phenotype. Altogether, this is how the mRNA sequences map to the antibody that will be created by our hypothetical B cell. B cell development occurs when a pathogen is first detected. A professional antigen-presenting cell, such as the dendritic cell shown here, first phagocytoses a pathogen and presents its antigens. This dendritic cell then interacts with the helper T cell that matches the antigen, which then activates an immature B cell with the receptor for the same epitope. This B cell begins proliferating, which is also called clonal expansion. Some B cells quickly turn into plasma cells and release antibodies to help the immune system. However, some of the proliferating B cells also go through somatic hypermutation. The variable region in the mRNA sequence that makes the receptor is mutated so that the receptor becomes even more specific. This new and improved plasma cell then also releases antibodies to help the immune response. Some cells, after somatic hypermutation, stay behind and become memory B cells. Memory B cells can recognize infections by the same pathogen next time and quickly respond. Another important cell in the immune system is the T cell that is responsible for both managing and killing pathogens with high precision and accuracy. A T cell begins as an immature T cell that has just been formed in the bone marrow. The T cell migrates to the thymus. Here, we show one of the lobes of the thymus. The T cell travels from the medulla to the lobe into the cortex. Along the way, the T cell develops and matures. In the cortex, it goes through certain forms of testing to ensure that the T cell has what it takes to fight against pathogens. After this, the T cell once again migrates down into the medulla and out of the lobe. As the T cell travels from the medulla of the thymus to the cortex, it experiences multiple important changes. When the T cell first arrives at the thymus from the bone marrow, it is a CD34 positive uncommitted progenitor. A lymphocyte expressing CD34 is simply a lymphocyte progenitor. However, once CD2 is expressed, it is committed to becoming a T cell. This cell is called a committed double negative T cell progenitor because although it is positive for CD34 and CD2, it is negative for two important proteins in T cell development. It eventually gains these two proteins, known as CD4 and CD8, and becomes an uncommitted double positive T cell. T cell receptors, similar to B cell receptors, have two chains. The chains can be either alpha-beta chains or gamma-delta chains. These chains are the next decision in the development of the T cell. If the beta gene is recombined first, the cell goes through alpha-gamma-delta recombination. If gamma-delta recombination occurs after this, the cell becomes a committed gamma-delta T cell. However, if alpha recombination occurs, it becomes a committed alpha-beta T cell. In the cortex of the thymus, the cells go through testing. The first phase is called positive testing because we're making sure that the T cells understand what a positive signal is. One example is that a T cell should recognize that a pathogen is a positive signal. Professional antigen-presenting cells use a protein called MHC class II to present antigens on their surface. All other cells use a protein called MHC class I to do the same if they're invaded by the pathogen. The T cell is tested by being shown both MHC2 and MHC1 proteins, as shown here. If the T cell better interacts with MHC1, it becomes a CD8 positive T cell. This is because CD8 is responsible for interacting with MHC1. Similarly, T cells that interact better with MHC2 become CD4 positive T cells because CD4 is responsible for interacting with MHC2. So when does MHC2 and MHC1 apply? Well, let's have a closer look. MHC2 is only used by professional antigen-presenting cells. This is because the cells must be able to phagocytose the pathogen and break it down before using MHC2 proteins. As seen in the diagram, the extracellular pathogen is taken in using phagocytosis. The vesicle is then merged with lysosomes to break down the pathogen. a brand new MHC class II protein, after being made in the Golgi apparatus, joins with the vesicles containing the remnants of the pathogen. The MHC2 protein then complexes with the antigen, which is a fancy way of saying they combine together. The MHC2 is then carried to the surface. Most normal cells do not need to eat pathogens because they have other work to do. Muscles need to contract, nerves need to send signals, and so on. Therefore, they only use MHC proteins when they get infected. For example, if a virus infected a cell, it would use a proteasome to break down the pathogen. The antigens created by breaking down the virus would then go to the endoplasmic reticulum and complex with a brand new MHC1 protein. This complex would then travel to the surface and get presented. Therefore, while MHC2 activates helper T cells to get the immune system started on hunting the pathogen, MHC1 tells the immune system that a cell needs to be killed because it is infected. If the T cell becomes a helper T cell, it further differentiates into two different types of helper T cells, class 1 and class 2. Class 1 cells are important mediators. They release IFN gamma to activate macrophages, LT to kill infected macrophages so that the pathogen becomes external, IL-2 to cause more T cells to develop and to activate NK cells, IL-3 and GM-CSF to cause macrophage differentiation, TNF-alpha to release T cells into infected tissue, and CXCL2 to attract macrophages to the tissue. Therefore, the class 1 helper T cell generally helps T cells and macrophages to arrive on the scene of inflammation and help. Furthermore, macrophages that get infected whilst trying to kill the pathogen are eliminated to avoid further infection in the body. Finally, macrophages and NK cells are activated to help stop the infection from spreading. Therefore, we can see why the helper T cell class 1 is so important. Class 2 helper T cells are important for activating B cells and helping them choose an isotype. The isotype is the alpha, delta, mu, gamma, or epsilon type of the antibody. This is accomplished using IL-5 and IL-6. Different isotypes combat different types of pathogens. Class 2 helper T cells therefore allow for a more efficient immune response. Furthermore, they also produce IL-2 to enhance B cell production. Negative testing ensures that T cells know what a negative signal is. For example, any part of the body should be negative, or else the immune system would end up attacking our own body. This is undoubtedly one of the most important parts of T cell development, as evidenced by the multitude of complications caused by autoimmune disorders. Basically, the T cell is presented with proteins from the body. If it treats any of them as a good epitope, it is marked for death. After this, the T cell is ready. It simply waits for an infection to occur with an epitope that matches its receptor. Let us assume that a pathogen infects the body that has an antigen that matches our T cell. First, a dendritic cell will phagocytose the pathogen and present it using the MHC2 protein. The first thing that the T cell must be able to do is recognize the antigen. If the antigen is not recognized, then the T cell cannot accurately find and kill the pathogens. Once the T cell receptor interacts with the antigen, the T cell receptor must also interact with the MHC. If the MHC class is not correct, the T cell will not be activated. Finally, the co-receptor must also recognize the MHC protein. After these three steps, one final interaction has to occur. The dendritic cell also has a protein called B7 on its surface. B7 interacts with CD28, a protein on the T cell, to send activation signals. Through all these interactions, a T cell finally gets activated to do its work. Thank you for watching this presentation. Here are the citations for all the images I used that were online and not made by me. Seriously, hope you enjoyed it.